Welcome all of you. In the last two classes, we started discussing about 2D NMR. I introduced what is a 2D, what is dimensionality in NMR, and how we get n time domain spectra. We do n dimensional Fourier transformation, and you are going to get a n dimensional frequency domain spectrum. This is what we saw. And we also understood how a pulse sequence is important. In each 2D experiment is defined by a given pulse sequence. There are n number of pulse sequences, design n number of 2D experiments accordingly. It has been developed. It could be both homonuclear and heteronuclear. It is one to correlate the information or to resolve the information. And accordingly, I said for homonuclear experiment, I gave examples of COSY, ROSY, TOXY, etc. In the result experiment, it is J-result experiment. Same way in the heteronuclear correlation experiments are there like HETCOR, HSQC, HMBC, HMQC, etc. And in each of these experiments, if a particular information is derived, there is a way in which magnetization transfer or the polarization transfer takes place from one spin to another spin during the mixing time. Accordingly, different information can be derived, and the pulse sequences are designed based on the dynamics of the pulse sequence, dynamic spin dynamics of the spin, how it behaves at different stages of the pulse sequence. We can retrieve, we know what information we are getting out of this, and this how we can even understand by pulse sequence, looking at the pulse sequence, what is that you are going to get in this pulse sequence. This we understood using product operators. Also, we know how to analyze the pulse sequence using product operators. That's how it is. We know all those things. And then I also said sometimes delays are important, pulses are important, where do you apply the pulse, which axis you apply pulse, which flip angle of the pulse, all those things matters a lot in each of these experiments. And let us say you do an experiment and they get the spectrum. How do you interpret the spectrum? If I have a peak, how it correlates the information in the, what happened to a particular peak? Let us say I have a peak at some 20 hertz or 30 hertz in the F1 dimension. And the same peak, if it has some frequency in the F2 dimension, how do you understand that? We discussed that several, like one or two examples we took. We said a peak which was evolving in the T2 dimension, I'm sorry, T1 dimension, not T2, in the T1 dimension, let us say at 30 hertz. If it also has only 30 hertz in the T2 dimension, then we say it remains unaffected. Whereas on the other hand, if I have a peak, let us say at 30 hertz in the T1 dimension, which has a peak at 70 hertz in the T2 dimension, then we can say in the T1 dimension this peak at 30 hertz evolved, and part of it magnetization is given to another spin, another signal which evolves at 70 hertz. As a consequence, part of it remains unaffected, which gives a peak at 30 hertz. And the one which has given a spin magnetization or polarization to another spin evolves at 70 hertz in the T2, and we get a peak in the T2 at 70 hertz. So there is a way we can interpret this type of 2D spectrum that we discussed, general way of interpretation of the 2D spectrum. And most important thing, we also understood how to what is the information content you get as you go to higher and higher dimensions. Higher the dimension, more time, it consumes more and more time. You can deal with bigger and bigger samples. Sensitivity also goes by root of measurement time. And we took a lot of examples, and I showed what happens, how much time it requires for a 1D, 2D or a 3D or 4D experiment, and also how it depends upon the size of the molecules. Varieties of information we discussed. That was basically the broad idea. All those things were extensively, all these were discussed in the previous course. This is for the benefit of those who didn't take my previous course. I am giving this. Now we will continue with the COSY sequence. This is a little bit commonly I have used experiment. Today we will start with a COSY experiment and understand what is a COSY, how it correlates the information. So we will start with this is a COSY sequence, and I take the example of scalar coupled 2-spin case AX. That is the example we take. Before going to that, we will take the uncoupled spin and see what is a coupled spin, etc. Right now we will worry about what is a COSY sequence like this. What does COSY does? COSY, the correlated spectroscopy is the expansion for it. It establishes connectivities between immediate coupled neighbors. understand immediate coupled neighbors. For example, I have a molecule CH2, CH3, some CH2X like that. Then if I take the Cauchy spectrum of it, it tells me CH2 is sitting next to CH3, and first this CH3, this is CH2 next, and this CH2 is next, like that. It gives the connectivity information between immediate coupled neighbors. Of course, you can go to not immediately, little longer also, that is a different, there are n number of variants of this experiment, but immediately you understand the Cauchy gives rise to connectivity information of the immediate partners, which is sitting next to which. That information you are going to get. And it basically it is a 2-pulse sequence, 2 90-degree pulses. That is a basic experiment, is one 90 degree here, it could be X or Y, here another 90-degree pulse. There are 2 90-degree pulses which is defined like this is the width of the pulse. And this is a T1 period in which it is a evolution of the magnetization takes place. This is a T1 period which is incremented systematically. You apply 90-degree pulse, of course there is no mixing period here, it is absent, but you can call this also as a mixing period, and then start collecting the signal. This is a detection period. Here you are going to collect the free induction decay, and here the T1 is incremented, you collect a pseudo FID and do the double Fourier transformation of it, you get the frequency domain spectrum, which in spectrum in 2 dimensions. That is what you are going to get, frequency in 2 dimension in Cauchy. Okay. Now, from our understanding, we know this is a preparation period, because this is the period under which we wait for the spins to attain thermal equilibrium. It comes to thermal equilibrium, and then this is the evolution where spins transfer their polarization, the information content of one spin is transferred to the other. That is what we are going to get the information. Here the polarization transfer takes place during the evolution period, and then we apply a detection pulse and start collecting the signal. Okay. Now, before going to the coupled spin, we will understand how does the 2D Cauchy spectrum look in an uncoupled single spin. When I have a single spin, there is no coupling at all, only one chemical shift. How does the Cauchy spectrum look? Now, this is the 2D spectrum. I do first 90-degree pulse tilts the magnetization to XY plane, that all of us we have been discussing for quite some time. This is what the first 90-degree pulse does, bring the equilibrium magnetization along Z axis to XY plane here. Then what will happen here? It will start evolving doing the free precision according to their offsets or according to their chemical shifts. They start evolving all the spins. And now we are dealing with a single spin, it will start evolving according to its chemical shift. After a time, let us say T1, what will happen to the magnetization vector which is in the XY plane? It starts moving, and how much it moves, the angle in which it moves is given by 360 star nu into T1. Because remember, we discussed this during the evolution of magnetization and the evolution of J coupling a lot. We wrote this equation and calculated how much is the rotation, how much is the different spin vectors moving when they have a different chemical shift with respect to the center nu naught, everything we have even calculated for 1 or 2 examples. So, after a period T1, the vector moves through an angle of 360 star nu of T1 degrees. Okay. Now, magnetization has both cosine component and the sine component. This is what we understood even from the product operators by 1 pulse experiment. Apply 90-degree pulse, you bring the magnetization XY plane, we understood immediately at the time t is equal to 0, magnetization goes to minus Iy, and then start developing both cosine and sine components. Oscillatory magnetization, it will be oscillatory components will be developed both along x and y, which we call cosine and sine components. We have discussed this a lot even during product operator formalism. So, magnetization is cosine component along y axis and sine component along x axis. Okay. Now, second pi by 2 pulse acts only on the y component. X component is not affected and continue to precess. Why x component is not affected at all? When I apply second after certain period during the evolution period, magnetization is getting evolved, and you apply after some time, you apply a 90-degree pulse. Now what is going to happen? The 90 pulse acts only on the Y component. What happens to X component? Continue to precise, it remains unaffected at all. That's what happens. The Fourier transformation of this one now is going to give me a spectrum which is a single frequency. And what is the intensity of the signal? That depends upon how much it has moved, with respect to nu z. This is given by 360 star nu T1. How many degrees it has moved, that depends upon. That's how the intensity you are going to measure. If the complete coherence is along one axis, where you have a receiver, you have maximum intensity. Then as it starts going dephasing, when developing other X and Y components, the intensity keeps coming down. That's what we have already seen. So this is the factor which defines the intensity of the spectrum. Okay. Now the Fourier transformation of this gives a single frequency, a spectrum corresponding to a single frequency, whose intensity is governed by 360 star into nu of T1. Okay. Now how intensity varies is given by this expression. We are taking the cosine component of that, cosine omega 0 star T1. Now initially let us say magnetization is like this, starts going like this, 0 red is and then starts going like this. It forms an oscillatory thing. Initially intensity is, let us say, negative, or you start like this, it goes like this. It starts intensity maximum, let us say, keeps on coming down, goes to 0 and then goes negative. And you continue like this. In fact, it undergoes oscillation. It is an oscillatory function. The amplitude varies in the time domain and it is a periodic function. It has a definite period, keeps on oscillating according to the period. This is what we discuss in the Fourier transformation, what is a periodic function, etc. Essentially, a pseudo FID is built in the F1 dimension like this. This is what we discussed while introducing 2D. At longer values of T1, what is going to happen? The signal intensity diminishes due to relaxation. It becomes very, very small, and finally there won't be any signal, you will collect only noise. So after which there is no point in collecting the signal. So the signal keep on decaying when you keep on increasing the T1 period. So you create a pseudo FID which is also decaying, collect the signal, and now you have a regular time period, you have a signal in the T2 period, that is a detection period, always that is a constant T2, T2 is constant, you have the FID there. Now we have a frequency data in one axis when you do the double Fourier transformation, that is T2, like similar to your conventional 1D NMR, and the time domain data in the T1, that is pseudo FID you have created. So you have a frequency. So Fourier transformation of the FID in the T1 dimension gives one frequency, and Fourier transformation in the T2 dimension gives you another frequency. Okay. So we have two things. I have time domain data both in T1 period and T2 period. Do the Fourier transformation of one time domain, you will get one frequency domain spectrum. Do the Fourier transformation, double Fourier transformation, both the dimensions, you get frequency domain spectrum in both two dimensions, you get a frequency spectrum. Okay. This is how the spectrum look. It is a single spin, no coupling, nothing. After double Fourier transformation, we get the signal where the two signals intercept. Remember, there is no coupling, nothing for it to transfer the magnetization, no other spin is there to interact. So let us say this is the frequency at which it is evolved in the F1 dimension, call it 40 hertz, let us say. It is evolving at 40 hertz during T1, it continued to remain 40 hertz in T2 also. It did not change at all. T1 if you go along this axis is 40, come along this axis it is 40. It did not change at all. There is no other spin to give the magnetization. Whatever the frequency it was evolving at which it was evolving in the T1 dimension, continue to evolve in the T2 dimension at the same frequency. So it is going to give a single peak. If you look at it, it is exactly on the diagonal. The diagonal peaks are the ones where the frequency of it, the evolution frequency in the T1 dimension and the T2 dimension both remain same. You understand? I have a peak, I have a spin which is evolving at a certain frequency in the T1 dimension. If that frequency remains same even in the T2 dimension, in a homonuclear experiment like this, Cauchy, you get that peak at the diagonal. On the diagonal you get that peak. That is a diagonal peak. Okay. So now I take the 2D spectrum of a two uncoupled spins. Both, there is no coupling at all. Very nice. I have one spin here, I have another spin here. There are two spins. There is no coupling. Let us say this is at 50 hertz, this is at 90 hertz. You draw a line here along this axis, draw a line along this axis. This is at 50, this is at 50. Draw a line along this axis here, this is at 90. Draw a line along this axis here, this is at 90. So in both the cases there are two uncoupled spins. There is no way they can transfer the magnetization between themselves. The peak which was evolving at 50 hertz during T1 continue to evolve at 50 hertz during T2. So that peak is coming on the diagonal. Similarly, the peak which was evolving at 90 hertz during T1 continue to evolve at 90 hertz during T2. It did not change, it did not give energy spin, it did not transfer its spin magnetization to any other spin. So it again comes at the same on the diagonal. So when there are uncoupled spins in the Cauchy experiment, the peaks are always situated on the diagonal. Important thing, please remember, if you have uncoupled spins, the peaks are always on the diagonal. What is the diagonal? This is the principal diagonal you have to consider, starting from left corner to the right corner. Not this one, this is anti-diagonal, this is the diagonal. Okay. So this is what is important when you consider Cauchy spectrum of two uncoupled spins. Now I took the example of two uncoupled spins and we wanted to see how the 2D spectrum look. This is how it comes. Both the peaks are on the diagonal. Okay. Now we will go further. What I can take this the projection of, I can plot on the F2 dimension like this. This is a projection. This is called F2 projection. Now both the peaks are coming here. This is called F2 projection. If I take the projection along this axis, this is called F1 projection. So F1 projection gives you spectrum which is projected along this axis, and the F2 projection gives you the spectrum which is projected along this axis. Okay. This is how basically the spectrum of 2D Cauchy spectrum of two uncoupled spins are there. Okay. This corresponds to nu x. Okay. This is nu x. It goes like this. This corresponds to nu a. This is also nu a. It goes like that. So the diagonal frequency, diagonal peak, if you measure the frequency, it corresponds to chemical shift of the individual spins. For this peak, this is the chemical shift. For this peak, this is the chemical shift. For uncoupled spins which comes on the diagonal, the frequency on the diagonal correspond to their chemical shifts, Please understand this. Now with this information, let us try to understand the Cauchy spectrum of two coupled spins A and X. Two spins are coupled. J coupling is present now. What will happen? During the evolution period, it evolves under chemical shift and also due to J coupling. Both are present now. Now very interesting thing is going to happen. Now we have a coupling constant JAX. After the initial 90-degree pulse, the magnetization associated with spin A will precess during T1 according to its chemical shift. Spin A has its own chemical shift, spin X has its own chemical shift. So it starts spinning in the T1 period at its regular chemical shift offset. Then it will produce a peak at the frequency A in both the dimensions nu A and nu A. That means both F1 dimension and F2 dimension, it is going to have a peak on the diagonal. It gives a peak here, gives a peak here. So that is what it means. That means the spin A is going to give rise to a peak which remains undisturbed both in the F2 dimension and F1 dimension and F2 dimension. This is F1 dimension, this is F2 dimension. So, I have a peak here which remains same. It is on the diagonal. Okay. This gives a diagonal peak. Next, what the second pulse does? That is what we understood for spin 1 case, single spin case. Second 90 degree pulse, here what happens is, it is does something different. In the second pulse, there was no coupled spin. It continued to evolve at the same frequency, gave diagonal peak in a single spin. Now, we have coupled spins. The second 90 degree pulse transfers part of its magnetization to the coupled X spin. A and X are coupled. So, A not only it is evolving during T1, when you apply a second 90 degree pulse, it transfers part of its magnetization to X spin. That is what is mixing. Here mixing is happening. In T1 period, both chemical shifts are evolving and J coupling is evolving. In the 90 pulse, mixing takes place and part of this magnetization gets transferred to coupled X spin. This is what is called polarization transfer. We discuss about this lot. The transferred magnetization will now, what will happen? It will persist in T2 dimension at the frequency of spin X. That is an interesting thing is happening. Untransferred magnetization, if there is no transfer magnetization, in the uncoupled case, both the time in T1 and T2 it was persisting at the same frequency. Now during coupling, it gives part of its magnetization to spin X. As a consequence, in the T2 dimension, this spin also persists at the frequency of the spin X. Imagine when we interpreted the 2D spectrum, I said there is a peak like this, one here, one here. It was persisting at this frequency both in T1 and T2. At the same time, it also had another frequency in the T2 which is here. So, the peak which was at, let us say, this frequency in the T1 evolved in this frequency. At the same time, it transferred its magnetization to other coupled spin and it evolved at this frequency in the T2 dimension. That is what it means. And this will this is going to give a peak like this, one on the diagonal which is evolving at its own frequency and another one which is evolving at the frequency of the coupled spin X, and this is what is called cross peak. Now, a peak in addition to chemical sheet of A also comes at the chemical sheet of X in the T2 dimension. This is called cross peak. Okay. Now, you may ask me what will happen to X spin. I was discussing about A spin. Identical behavior. X spin also behaves like A spin. That means it transfers, it evolves, let us say, its own chemical shift frequency in the T1 period and also in the T2 period, gives us a diagonal peak and gives part of its magnetization to spin A and evolves in the T2 dimension at the frequency of spin A, at the chemical sheet of spin A. You understand? Now, two things you have to understand. A spin was coupled to X. A will evolve in the T1 dimension at its own chemical shift frequency. During the mixing, it gives its energy or polarization to X spin. So, it evolves not only at its frequency, but also at the frequency of X to which it is coupled to. Identically, spin X evolves during T1 at its own chemical shift frequency. In addition to that, because it is giving its polarization to the coupled spin A, in the T2 dimension, it evolves also at the chemical sheet of the spin A. Essentially, you are going to get a spectrum like this. One here, one here. This will evolve at the chemical sheet here, this will evolve at the chemical sheet here. Okay? And it also gives a diagonal. Now, now we are talking about X spin, it should have been XX. not AA, I'm sorry. Sorry for the mistake. It should be nu x, and also cross peaks at nu A, nu x. You understand? So this is what is happening. X spin behaves identically like A spin. Like A spin gave a diagonal at nu A and nu A and nu x cross peak. X spin gives diagonal at nu x and it is going to give cross peak at nu A and nu x. That's what happens. In effect, the precision of the magnetization during the T1 and T2 period is no longer the same frequency. The magnetization precises at two different frequencies. Its precisional frequency in the T1 is different. Its precisional frequency in the T2 period are different. So, as a consequence, 2D spectrum contains diagonal frequency of nu A and also cross peak at nu A and nu x for both A spins and X spin. Please understand, the precision frequency of the magnetization during T1 T2 periods are no longer same. The 2D spectrum contains a diagonal signal nu A and nu A for spin A, nu x and nu x for spin X, and nu A and nu x and nu x and nu A are the cross peaks at their respective chemical shifts. So, X spin also gives a cross peak and also a diagonal peak. The diagonal peak has a very interesting structure. It has a fine structure of 4 peaks. Why? Because it is J coupled. Okay. Diagonal peak is not just evolving at the chemical shift frequency. In fact, I did not tell that because I did not want to bring in this coupling till now. Now I am bringing in the coupling. It is not evolving at nu A. It is evolving at nu A plus half JAX and nu A minus half JAX in the F1 dimension. Similarly, in the F2 dimension, it evolves at nu A plus half JAX and nu A minus half JAX. That means, in both F1 and F2 dimensions, you get doublets. So, I am going to write a diagonal peak like this. If I get a 2D spectrum, I am going to get a diagonal peak like this. I have a 2D spectrum, I am sorry. I have a 2D spectrum here. Let us say I am concentrating on the spin A. Earlier I said it is resonating at the chemical shift of spin A. No. What happens is it will have one peak here, one peak here. Half this is J A, half of nu A minus half JX. Here is nu A plus half JX. Similarly, you have a peak here and here. So, in reality, how it comes is it comes like a square. Okay, I will tell you it comes like a square. Maybe I have the drawing, I will show you. It comes like a square. What is going to happen is it comes like a square. Okay. So, we will see that for this one now. For spin 1, the diagonal phase are in phase doublets. We discussed in phase, anti phase, in phase dispersive, in phase absorptive, everything. They are in phase doublets. Both are in phase doublets in the T1 dimension. Similarly, in the T2 dimension or in the F2 dimension also, it is in phase doublet. Both are separated by J. Interestingly, both are separated by J. Okay. So, for spin 2 also identically, the diagonal phase are in phase doublets, both in F1 and F2 dimensions, centered at the frequency nu 2. Okay. So, the cross peaks also will have a similar dimension, similar structure. In the sense, cross peak arise from the transfer of antiphase coherence to antiphase coherence to remixing. That is what we discussed. I told you how magnetization transfer, coherence transfer takes place in the inept experiment. In product operator also we understood it. This J coupling is responsible for creating the antiphase coherence, and this is responsible for transfer of magnetization from one spin to another spin. That is what we said. So, because of the transfer of magnetization, the cross peaks are antiphase in character. That means if one signal is positive, other is negative of the J split doublet. This is a doublet split because of J coupling. In that one is positive, other is negative. Okay. The cross peaks are antiphase in character in both the dimensions, not only in one dimension F1 and both in F2 also. So, that is why similarly cross peak has a fine structure, 4 peaks in a square pattern, cross peaks in the F2 dimension are doublet that are 90 degree out of phase with respect to F1. Similarly, in the other dimension also it is a doublet and is out of phase. Each of them, each one is a positive, the negative peak antiphase doublet, and the separation in each case is always a J. But you do not try to do the phase correction here. You cannot do that. I remember I told you when we were talking about in phase, antiphase, and all those things, dispersive, absorptive. I said dispersive, absorptive, you can make the phase correction, but in phase, antiphase, you cannot do that. If you make in phase, antiphase, again other will become antiphase. So, you cannot correct the phase of these two. So, it is not possible to correct the phase of this cross peaks. As a consequence, the spectrum of the Cauchy is always represented in the magnitude mode. The spectrum is always represented in the magnitude mode. That is the important thing. Okay. So, we will diagrammatically see how this diagonal peaks and cross peaks comes and etc. Okay, it takes some more time to explain. It will take at least more than 5 minutes. What I am going to do is, since the time is getting up, I will stop here. But today what we discussed is, we discussed about the simple example of a 2D Cauchy sequence. Cauchy is a correlated sequence, correlated spectroscopy. It is nothing but a simple 290 degree pulse sequence with a T1 and T2 period. T1 is systematically incremented. Do the double Fourier transformation, you are going to get a frequency domain spectrum. And we took the example of uncoupled single spin. If it evolves at a particular frequency in the T1 dimension, it evolves at the same frequency in the T2 dimension, because it is not coupled to any other spin. There is no way it can give the transfer the magnetization because of the mixing during the second pulse. So, it always gives rise to the same frequency in both F1 and F2 dimensions. So, if you plot it in the 2D spectrum, in the homonuclear 2D spectrum, this gives us a single frequency appear at this diagonal, on the diagonal of this thing. So, exact on the diagonal you get a peak whose frequency is exactly same on both F1 and F2 dimension. And then we took the example of two uncoupled spins. It behaves same. That is what we said. Two uncoupled spins also behave same. There is no exchange of magnetization, transfer of magnetization between two spins, 1 and 2 or X, A and X, whatever it is. But then both of them give rise to single peak exactly on the diagonal. That means each of these uncoupled spins continue to evolve in the same frequency both in F1 dimension and in the F2 dimension. Then we went to the example of two coupled spin. There we understood, in addition to individual chemical shift offset, there is also evolution due to J coupling during T1. When you apply a mixing pulse, there is a transfer of magnetization from one spin to another spin. Antiphase coherence transfer takes place between two spins. As a consequence, what is going to happen is, the spin A, which was precessing at one frequency, will also give its magnetization to spin X, and then it starts precessing at its own, in addition to its own frequency, its frequency of the X in the T2 dimension. Same thing for the X spin. So, as a consequence, in addition to its own frequency, which is at the diagonal, whose frequency of evolution remains same both in T1 and T2, it also gives a cross peak. Same thing happens for the X nuclei, X spin. That's what we discussed. But we also said it is not exactly at the sum of the chemical, at their respective chemical shapes. Because of the J coupling, there is a splitting, and it is coming, you will have doublets in the F1 dimension, you have doublet in the F2 dimension, especially for the diagonal, both two doublets, if you say they are in phase, they are positive in phase, absorptive. Whereas cross peak, because it is antiphase coherence to transfer to antiphase coherence, they have antiphase in character. So, you cannot correct the phase of that. So, as a consequence, you will represent the Cauchy spectrum always in the magnitude mode. This is what I said, and we will see the sign of the diagonal and cross peaks, how the spectrum looks, etc. in the next class, and take 1 or 2 examples of it, try to analyze the spectra and go to other 2D spectra later. So, thank you. I am going to stop here. We will meet again in the next class for further discussion. Thank you.